Hello, everyone. Good afternoon. Thank you so much for joining us. Could I just get a thumbs up from folks if you can hear me okay? Excellent. I don't want to get too far into things without checking. My name is Kim Weed. I'm a teaching assistant professor in the Department of English and Comparative Literature, and I'm the co-director of the Hive Lab, which is organizing this series of health humanities grand rounds events with folks at UNC, at regional institutions, and beyond, who are interested in and focus on questions about human health and well-being that have a humanities perspective or methodology. I want to thank you so much for joining us today. And of course, to our sponsors, the Hive Lab and Carolina Seminars, who make this series possible. Before we actually get started with the talk itself, I'd like to just go over a few logistics. We ask that you keep your microphone muted throughout the talk, just to minimize distractions. And then you are, of course, welcome to keep your camera on to give Dr. Winstead a bit of an audience. It can be challenging to present to a blank screen. And then today for Q&A, we are going to use the chat function to help moderate questions. So you have options. You could either type out your question altogether, and either Irmak or I will read those to Dr. Winstead. Or if you prefer to voice your question yourself, you can type into the chat that you have a question, and that will hold your place in line when we arrive at the Q&A. Finally, this talk will be recorded, and we will eventually get it posted to the Hive Lab website and the Hive Lab YouTube channel. And you're welcome to check out past speakers for this Grand Round series. Okay, with logistics out of the way, it is my absolute pleasure to introduce you to Dr. Mike Winstead, an assistant professor of pediatrics, hematology, and oncology at UNC, who balances clinical expertise in hemoglobin disorders with research interests in stem cell transplantation, both in terms of the immune system function and efficacy, but also, as we'll hear soon, in the broader cultural implications of how the field of stem cell transplantation understands race, ethnicity, and heredity. I had the absolute pleasure of meeting Mike over the summer over coffee to discuss some of his growing interest in health humanities. And when he described his thinking about how stem cell transplantation science engages with ancestry, I immediately thought of this series as a venue to share some of that work. I'm just so thrilled that he agreed to share his work in progress with us today. So please join me in welcoming Dr. Winstead to Health Humanities Grand Rounds.
Thank you so much, Kim. This is a huge honor. Let me just start sharing my screen. So again, my name is Mike Winstead. Let me just, there we go. All right, is it in full screen and everything? All right, perfect.
It is,
so I'm
yeah.
going to be talking about the concept that I'll call the transplanted self and the intersection of race genetics and pragmatism in stem cell transplantation. It gets to be a habit as a physician that you put a disclosure slide at the beginning. So I don't talk about any branded products, and of course I'm not receiving any corporate sponsorship for this talk. Also, I do think it is important to disclose there aren't any graphic medical images in this presentation. Doctors can be a little bit callous about including startling images, and I did not include any of those. What I do have is a nice picture of bone marrow to get started with, because that is where we're going to be spending a lot of this talk. The bone marrow is a liquid that's contained inside of the bones, and it functions as an organ that produces blood. Specifically, there are stem cells in the marrow that produce blood, and you can transplant those stem cells from one person to another, which is called a bone marrow transplant or a stem cell transplant. I will use those terms interchangeably. Bone marrow was the original process for doing this, to take one person's bone marrow and put it into another person. There are other ways of getting stem cells, for example, mobilizing blood-producing cells into the bloodstream and collecting them that way. Or newborn babies actually have a surprisingly high concentration of stem cells in their umbilical cords. And so at some hospitals, parents can donate the baby's umbilical cord for stem cell transplantation. After infusion into a person's bloodstream, the blood-producing or hematopoietic stem cells will simply find their way back to the recipient's marrow cavity and begin making blood. And often the first conversation I have with a family is that I'm not a surgeon, and what I'm proposing for their child is not a surgical procedure. It's a lot more in line with a blood transfusion. This is one of the kind of wonderful things about the field. Why would somebody need a stem cell transplant or a bone marrow transplant? Across the board, the most common indication is leukemia and lymphoma. This is data from 2018, collected from both adults and children. There are rarer indications like failure of the bone marrow to produce cells, thalassemia, sickle cell disease, immunodeficiencies. These are not common things in childhood, but they are relatively common indications for transplant in children compared to adults. This is also, I should probably state at this point, there's a separate area of transplantation called autologous transplantation, where a person donates stem cells to themselves and then receives them after some kind of treatment for an autoimmune disorder or multiple myeloma, for example. That's a separate topic, since the genetics of that are obviously not applicable. The person is donating to themselves. What we're talking about is the terminology is allogeneic transplant, where there is a donor, second person whose bone marrow and stem cells are going to be infused into the recipient. So in the process of producing blood, the stem cells in the bone marrow also produce the immune system. Now, I think our introduction to stem cells is often this concept of a pluripotent stem cell that can turn into anything in the body. I know very little about pluripotent stem cells because I work with hematopoietic or blood-producing stem cells. Broadly speaking, hematopoietic stem cells produce a myeloid lineage, which goes on to make red blood cells, platelets, and granulocytes, which are one type of white blood cell. And then it also produces a lymphoid lineage, which produce the lymphocytes. And collectively speaking, the granulocytes and the lymphocytes constitute our immune system as we currently understand it. So when you receive another person's bone marrow or another person's blood-producing stem cells, you receive their immune system, and that is where the real challenge of transplantation comes in. So the immune system is an interesting concept. The idea of immunity or exemption, literally exemption from infection, have existed throughout recorded history, and the recognition that people who developed an illness with a very characteristic appearance, like measles, for example, would only have that happen once in their life, was recognized for a very, very, very long time. And it long preceded even the idea that illnesses were transmissible from one person to another. Immunity is a social concept because the recognition of immunity or exemption from infection occurred within this somewhat complicated understanding of how diseases were experienced in society, and especially how people caught diseases from their environment and in the way of thinking until fairly recently. Now, more recently, you can see this illustrated in something like this medical illustration from 2016 in a journal called Blood Review that is talking about two different cells of the immune system, the T cell, which they liken to a contract killer, and the natural killer or NK cell, which this author likens to a border patrol agent. And as we're talking about some of the concepts at play here, I think one of the challenges that you run into in the medical literature is that sometimes the full implications of a metaphor are not explored, and the reverse of this statement that a border patrol agent is like the immune system of a country is probably not what was intended here, but it still is pretty easy to come across. Within medicine, this concept of immunity or the function of the immune system is articulated in terms of an immunological self. And the term that was coined many years ago in the 1950s and early 1960s was that the immune system has to distinguish between a self and a non-self, and that the fundamental operation of our cellular immune system is that distinction. The person who coined that term actually tried to take it back later. He found it to be a little bit too loaded of a term, but by then, because probably it was such a loaded term, it had kind of taken on a life of its own. The irony here, when we talk about the immune system being a border patrol agent and this concept of a rigidly bounded immunological self, is that the idea of having an immune system and this idea that there is a group of cells in the body whose responsibility is to prevent infection and maintain the integrity of our interface with the outside world, a lot of that knowledge and research emerged from the field of organ transplantation, and before that, the technology of skin grafting. And the goal of those technologies is not to maintain a boundary between the self and the quote unquote non-self. It's to allow the seamless unification of two different people. And so medicine has been attempting to cross this border for as long as we have been establishing that this border exists. Within stem cell transplantation, one of the key things that we are shooting for in a stem cell transplant is to have a donor immune system that functions normally within the person receiving the transplant. In contrast to someone who receives a kidney, for example, or a lung transplant, a person receiving a stem cell transplant probably will not require lifelong immunosuppression if everything goes well. Eventually, the recipient's body retrains the immune system to function correctly and not cause problems, which we'll talk about in a moment. As a consequence of that, a person goes through their life after a stem cell transplant with their blood containing their donor's DNA. And this is one of the things that we talk about before a transplant, and people have a fairly consistent way of responding to this. One of the things that tends to come up, especially if it is a family member donating, is if I commit a crime, could I leave my family member's DNA at the crime scene? But the other one that comes up very frequently when it's an unrelated person that they've never met before who's going to be donating marrow to them is sort of a joking question about whether they're going to take on the traits of that other person, whether they're going to start speaking a different language if the donor is overseas, if they're going to, have a change in their hair color or their accent or other things like that. And it's a joke, but it gets at this idea that sort of our bodily integrity, and especially the DNA that's contained in our blood, has a lot of meaning to us. And while we can accept intellectually that this is a technology that works and that is rational, there's still a lot of meaning embedded into it that comes across as you're doing this process. So that combination of the donor and the recipient at a genetic level, the term that we have for that is chimerism. And this is the ancient Greeks loved chimeras, which were monstrous creatures that had parts of multiple animals all grafted together. So this is a lion with a goat head and a snake for a tail. It's not a great term, obviously, for that reason, although the other place where the word chimerism is used, as is the word transplantation, actually long preceding its use in humans, is in botany. And so this is a chimeric dahlia made from grafting together two different plants to produce this combination. So as we're attempting to produce a chimeric person, or to have a seamless unification of the person receiving the transplant and the cells that they receive from their donor, it's important to talk about the mechanics of immunological selfhood. So as we currently understand it, a group of lymphocytes called the T cells are most important in the process of transplantation. The T cells interact with other cells in our body, for example, the cells that constitute our skin. And that interaction is mediated by a cellular surface marker called the major histocompatibility complex, or MHC. There are actually two of these, but we don't have to go into that level of detail. If a cell becomes abnormal, for example, because of a viral infection or because it is developing into cancer, it will interact with the T cells differently through MHC. And at that point, the T cell becomes activated and eliminates the infected cell. After a stem cell transplant, this process continues as it would otherwise, with the significant wrinkle that the T cells are now derived from the donor's stem cells or the donor's marrow, and they are interacting with, for example, the skin cells of the person receiving the transplant. And so if, for some reason, the recipient MHC, or major histocompatibility complexes, trigger the donor T cell, the donor T cell responds in exactly the same way it would to an infection and eliminates the activating cell. This reaction is called graft versus host disease, or GVHD, and it can be very, very severe or life-threatening after a bone marrow transplant. Also, in the grand scheme of things, again, our goal is to have a functioning immune system after transplant, and so graft versus host disease would significantly interfere with that. So a lot of what we do in preparation for a transplant, and the place where genetics enter into this, is in the process of matching the immune systems of the donor and the recipient and trying to prevent graft versus host disease. So those MHC, or major histocompatibility complexes that I mentioned, are coded by genes called HLA, or the human leukocyte antigens. And those, as with all of our genes, are inherited from our mother and father. We have two sets of these genes. So a person with the same biological mother and father has a chance, a 1 in 4 chance, of inheriting the exact same sets of HLA genes as we have. And that allows for what's called matched sibling donor transplantation. So when two people who have inherited identical genes from the same parents donate to each other, it's a matched sibling transplant. This was for a long time the main way of doing bone marrow transplantation, and as a result, the kind of safest approach to bone marrow transplantation or stem cell transplantation privileges a nuclear family structure in which you have parents and biological children of those two parents. It's implicit in the field right now. Now, also, parents and children will typically share half of their HLA genes, since they are passed from generation to generation. I say typically because exceptions can occur, but you would expect that a parent and a child would be 50% matched to each other, which facilitates something called haploidentical transplantation. Now, if it sounds like the difference between 50% and 100% is probably quite significant, it is. And this is a more risky approach to transplantation, but it's also much more feasible, since most people, not all people, but most people have one biological parent in their life, and or at least one sibling, and your siblings are more likely to inherit one set of these genes from their parents than both sets, if that makes sense. Then the other area where we can get a stem cell transplant is that unrelated individuals can have identical HLA genes, which facilitates unrelated donor transplantation. And the reason for this, in kind of technical terms, I suppose, is that HLA genes exist in linkage disequilibrium. This is an illustration of linkage disequilibrium, where on the right-hand panel you have all of the shapes, all of the circles are black and all of the squares are white. On the other side, you have a mix of black and white circled in squares. So on the right hand, you would have linkage disequilibrium, where shape and color were quote unquote co-inherited with each other. Another example would be eye color and hair color, which are things that you would presume that someone who has brown eyes like me would also probably have brown hair like me, and that someone who has lighter colored eyes would have lighter colored hair. And what I just did there is actually one of the other problems that creeps into these conversations, which is that we can slip very easily between talking about observable traits in people and talking about genes, and then going in the other direction, we slip very easily from talking about genes to talking about traits, to talking about the people who exhibit those traits, to talking about the social structures within which those people exist. And that slippage is probably inevitable to some extent as we're trying to implement any kind of genetic information in the real world. But we do it without even really recognizing it. And a lot of that has to do with the fact that the idea of heritable traits, or broadly speaking, the field of genetics, preceded the recognition of genes or the discovery of DNA. Like the idea of immunity, the concept that people and families are similar to each other has been around for a very, very long time, understandably. And I think we also all recognize that the similarities between people have been used for justification of any number of social organizations over the years. That's being a bit euphemistic, and I apologize for that. So this is where the process of transplantation begins to interact with a much larger society, is in the process of unrelated donor transplantation. So this graph, I edited this graph to make it more legible, but the information is the same. Until very recently, the National Marrow Donor Program, which was the United States organization to match people with donors, had this image on their public-facing website, which along the x-axis here has a number of ethnic or racial groups, and on the y-axis has the likelihood of finding a match an HLA matched donor. The blue bars are a perfectly matched donor by the technology of the time, and the yellow bars are a partially unmatched donor. Now, ironically, I've been using this slide in talks for many years, and when I went to get it from the NMDP website for this talk, it is no longer there. They've actually replaced it with a piece of boilerplate that says, currently the chance of having a matched available donor ranges from 29 to 79%, depending on patient ethnic background. And that low number there, which I've highlighted, the people with, or the groups on this x-axis with the lowest likelihood of finding a donor are classified as African American, African, Black Central or South American, Black Caribbean, and then Southeast Asian, Hawaiian, Pacific Islander. And that's a very strange combination of terms. You have, people from a continent alongside people of a demographic group within the United States, along with, a different kind of demographic terminology that's applicable to other parts of the Western Hemisphere. In a reference, this data comes from a very influential New England Journal of Medicine article from 2014. This is the same data in a slightly more eye-searing format. The takeaway of this article was that many people don't have optimally matched donors, which is a public health issue. And they go on to say that the reason for that is that the levels of polymorphism of the HLA genes are high, meaning there's a lot of diversity in these genes, and that allelic variation is population specific. And population is a very significant word in there, because the way that we define populations is in some ways at the heart of this topic. So in citing their development of demographic groups for this data set, the authors of that study referenced a report from the 2010 census. And so there are a couple of things that are interesting here. One is that the census does not have 21 different groups, that has, 7, I believe. The other thing that's very important is that there was a group on the census of people who identified as 2 or more races, and that is a group that is absent from the slide that I showed a moment ago. And so in addition to privileging nuclear family structures, transplantation tends to privilege people of singular or identify as singular race or ethnicity. In larger terms, this is part of that slippage that I mentioned a little while ago, is that the rationale behind grouping people isn't based on something genetic, obviously. It's based on the demographic systems that already exist in society. And these demographic and political designations of people that are important and that are real also inform our genetic and public health and sort of biological understanding of human diversity. And the implications of that, which is what I'm going to spend the rest of the talk talking about, haven't really been explored in the level that they probably could be. So if you go to the medical literature, the public Medline database, and look for the diversity of HLA genes, there aren't all that many reports on it, but there was one from 2003 where they looked at regional differences in people identifying as African American and their HLA genes. And they found that depending on the region of the United States where a person lived, the people in different regions were more similar in their HLA genes than people from different regions. And the authors of this study suppose that it may be due to different ancestral origins, migration, and admixture. And it may be important to consider at sort of a public health level. That term admixture is a very strange and striking one. The places where you find admixture in the medical literature are conversations about race or ethnicity, especially in terms of transplantation, and in the literature on preparation of medications for injection and mixing medications in a pharmacy. This concept of literally adding one substance to another is an evocative metaphor for better and worse, and it's a surprisingly tenacious one. And of course, it's not socially value neutral. So last year, a publication in Frontiers in Immunology looked at the chances of finding a donor based on the level of Africanness of the person looking for a donor. And this was a study in admixed Brazilians. So looking at demographics, Black versus white, and then these authors quantified people's Africanness at a genomic level and at the level of the MHC or HLA genes. Um, there isn't a discussion in the study of, the kind of fraught history of quantifying people's Africanness. And I think in even larger terms than that, the idea that when you're talking about admixture, it isn't this value neutral thing. It's really the amount of Africanness that a person possesses, or the amount of Africanness that has been, quote unquote, added to a sort of base material, is obviously very problematic, and it gets at the challenge that medicine runs into trying to navigate these spaces, because on the one hand you have this genetic information, on the other hand you have the social structure within which you're processing that genetic information, and the effort to resolve those two things ends up with this rather strange example where you are trying to biologically quantify something that is fundamentally social in nature without acknowledging that that's what you're doing. Another article from the journal Genetics in 2015 asserts that population level genetic models show a decrease in human genetic diversity at increasing distance from Africa, with the caveat that admixture, there's that word again, between populations and changes in population size also affect genetic diversity. And this publication it has this illustration in it, which I actually saw presented at a transplant conference when I was in training, which shows, human genetic diversity basically as these colorful bubbles. And as people migrate along these arrows across the world, their genetic diversity is kind of deposited in one region or another and decreases as people move along. The implication here, and what many of us take away as we're learning the process of transplantation, is that the genetic diversity that we're encountering as we try to match people with their donors is the product of human migration that happened tens of thousands of years ago. And then ... here we are in 2021 trying to do transplantation. There are a lot of assumptions missing. I would also like to add that this is not something that's confined to the medical literature. You can find essentially the exact same diagram on the 23andMe blog. So this concept of where our genetic diversity came from and what population genetics mean, and specifically the population genetics happened when people were crossing the Bering Land Bridge 15,000 years ago. This is something that operates in society at large. It's not a buried concept. There's a very obvious thing missing from all of this, which is, what you could say is the source of the admixture, in which would be more bluntly described as the Middle Passage. The genetic diversity of the Western Hemisphere, and honestly the world at this point, was tangibly impacted and probably defined by the slave trade and the colonial project. And this is something, if you search for slavery and bone marrow transplantation, this is a completely unexplored area. People simply don't talk about it. And there are a number of reasons why that might be. One is that the politically charged nature of this topic allows for room for people to manipulate biological and genetic data in a way that supports an antisocial project. But I think the downside of kind of leaving this concept of diversity back in the far distant past at the level of our ancient ancestors, migrating, in the Iron Age is that we overlook the things that we just speculate about in the medical literature, and especially that we overlook the amount of research and knowledge that exists into the origins of genetic diversity as we encounter it today. On top of that, even in the more recent past, when you have someone saying that migration patterns could have influenced the genetic diversity of people in the United States, those migration patterns were not coincidental. There was a very well described and very well studied pattern of migration within the United States as a result of laws on segregation. And likewise, as you talk about the quote unquote admixture of people in Latin America, that was similarly driven by by social policies that promoted the forced intermarriage of indigenous people and enslaved people with the colonizers and the enslavers. So these are socially embedded concepts that we find manifesting themselves through population genetics, and we're not clear, I don't think, as a field or even as a society on what the full implications of that are. On top of that, outside of the United States, we coordinate with other donor centers around the world to identify donors internationally that could donate to our patients. And this map from the NMDP of donor centers around the world has a rather noticeable hole in it around the equator. One of the many peculiarities of this is that there are 47 transplant centers in Europe, and they counted, I believe, Egypt as part of Europe for this, because there are transplant centers along sea here, and there are only 2 transplant centers in sub-Saharan Africa. Just in case this wasn't clear enough, if we plot these same stars roughly on top of a map of economic data, it's pretty obvious where these imbalances come from. As with everything else that we're talking about, these aren't coincidences. It's not a coincidence that the healthcare infrastructure of Central Africa cannot support transplant donation centers. That is the result of the exact same colonial project that produced the slave trade and that has produced the concept of genetics that we're currently grappling with. So the medical procedure that we're doing when we transplant one person's bone marrow or one person's stem cells into another person's body, that is happening at this biological and social intersection of forces throughout history that include, this broad concept of ancestry, or where we come from as individual people, but also the history of slavery, the history of colonialism, and the history of segregation within the United States. And this intersection has terms that are genetic, it has terms that are cultural, and it has terms that are economic, and these are all overlapping with each other, which is to say that we have this overdetermination of the outcomes of transplantation, which is where we're going to be going into next. And within medicine, this manifests itself in a number of different ways, and you encounter a lot of frustration with the challenges of identifying donors and getting people to transplant, and specifically frustration with the fact that those lapses in transplant availability tend to be concentrated in the most vulnerable members of our society, and those are not coincidental things. The trouble is that the language with which we articulate those discrepancies can come across in a somewhat counterproductive way. And so this was another a very good article from 2019 about the difficulty in matching Black people in the United States with marrow donors. And these authors said that the efforts to improve minority recruitment are unlikely to meaningfully address transplant disparities because the population is becoming more diverse. A bit of a loaded statement there. At the risk of editorializing, it would be more accurate to say that this transplantation science evolved within a sort of artificially homogenous society in the mid-20th century United States, that the privileging of nuclear family structures, the privileging of singular racial identity, and the implicit privileging of whiteness were all things that evolved from that society at that time, or from our society at that time, and that we are simply dealing with the long-term ramifications of a technology developing under those artificial conditions. At the same time, the takeaway of this paper, and the reason that they wrote it, is to say that there's no disputing the ethnic and racial barriers to transplant, and that it's essential to find alternative graft sources to make sure that everyone gets access. So the approach that we tend to take in medicine is to identify a tangible problem, in this case, that there aren't enough donors for people who are not white, and then to figure out a technological solution to it in order to transcend that problem and move on to the next one. The trouble is that you don't necessarily grapple with the nature of the problem that you are solving, and it just allows that larger question to proceed. So when we talk about this transplant itself that I alluded to in the title, the transplant itself is this physical unification of the donor and the recipient. And there are any number of things that you could talk to that inflect that process. There are things that happened a long time ago, like population genetics. There's this general concept of familial ancestry. There's the availability of registries or the size of registries, which is influenced by public policy. You have the availability of social support and income for the person receiving the transplant. You have the person's family structure. You have their physical and mental health, not only of the recipient, but of the donor. And so at the risk of oversimplifying, we could kind of cluster these things into a big picture concept of ancestry, and then also this concept of history. And if you ask me the clear distinction between those things, I would not be able to give it to you, but I'm sure we could debate that. They're interchangeable things. In the genetics literature, ancestry generally refers to a person's distant past background that can be identified genetically, and history is sort of a narrative process through which their existence is understood. But those aren't, I don't think those are distinct concepts. Our sense of history influences our sense of ancestry and vice versa. Those arrows crossing the Bering Land Bridge weren't put there by genetics. They were put there by our kind of shared understanding of where we came from as humans. So there's this kind of complex intersection at which a person receives their transplant and becomes the chimeric person that we're all striving for. And that is all happening against the backdrop of this much more complicated social and cultural history. When I was making this slide, the question popped into my mind: where does medicine actually fit into this? And like, which one of these boxes or arrows is medicine? And I kind of decided that it's these blue arrows here that are connecting the donor and the recipient. And that's really, by design, the extent of medicine's engagement with this entire process. And that's the challenge that I'm hoping that future generations will be able to address. So one question that I received as I was preparing this talk is, how does this unfold in practice? What does this look like for a person who's receiving a transplant? And I think that can be quite illustrative. So this is all obviously HIPAA compliant. There's no identifiable health information in here. But I took care recently of a 15-year-old girl who was diagnosed with acute myeloid leukemia, and her leukemia contained a mutation associated with a very high likelihood of relapse. She received 10 weeks of chemotherapy to get into remission, and she was then a candidate for a stem cell transplant, and she was Black. So we met with the family to see if there were any siblings who were potentially eligible to donate marrow. She had an older sister, but her older sister had a different father, so she would not have been eligible to donate. We looked into the registry to see if she would have received, if she had an unrelated donor available. There were no HLA-matched adult volunteers. We checked for cord blood units, which sometimes you can find an umbilical cord blood unit that's available, and she did not have any that were adequately matched or that had an adequate number of blood-producing stem cells for her. So the two conventional approaches to transplant were not available to her. So then we moved on to the process of a mismatched transplant. We reached out to her mother, but they were estranged, so her mother was unable to donate. She lived with her father, who was haploidentical or 50% matched as expected. So she received a transplant, which consists of high-intensity chemotherapy, an infusion of bone marrow into the bloodstream. And then in her case, she received additional high-intensity chemotherapy after transplant with the goal of preventing graft versus host disease, along with a course of immunosuppressive medications, likewise to prevent graft versus host disease. There were a number of obstacles that she ran into. She had some fairly severe depression that developed after her transplant, and it manifested in inability to take her medication, which was a problem that had to be navigated. And then as we were working through that problem, she came into the hospital unable to breathe, required intensive care, mechanical ventilation. We identified that she had a severe fungal infection, which can happen when a person's immune system is profoundly suppressed. We asked that her home be evaluated for toxic mold before we sent her home from the hospital. Meanwhile, she recovered. And sort of at the juncture of these things, her recovering from her illness and her home being evaluated, her landlord evicted her family from their home. She became homeless. This was at the height of the COVID pandemic. So we were able to find some temporary housing, but while she was staying in an extended stay hotel, she developed COVID-19 infection. And as she was recovering from that, we identified that she'd developed graft versus host disease of her lungs, which is ultimately a life-limiting condition. She has since reconciled with her mother and moved in with her mother out of state. And the last update I got is that she's actually doing quite well. But this was still a very bad outcome for her. She had a miserable life for about 2 years after her transplant. And in the long run, her outcome is going to be very, very bad. And the frustrating thing about this is that when you take a step back, you realize that, I think it was Frantz Fanon who said that that Blackness is overdetermined, and people have expanded upon that in a number of ways to say that, when a catastrophe befalls a Black person in the United States, you can point to any number of factors that contributed to it, and that process of overdetermination, that you have, financial instability is not independent from the inability to find an HLA matched donor for your transplant, and none of those are independent from the kind of resources that the state makes available to children that don't have a place to stay. You can point to this kind of conglomeration of factors, and these are all embedded within that larger structure that I talked about. And it becomes very frustrating to go through this with a family. The part of the reason that medicine tends to be, in my opinion, very focused on the practical and concrete problems, or this habit of what I'll call medical pragmatism, is that they get your attention and you have to deal with them. And you can point to the individual situation as unique, and taking a step back from it can be very challenging. And so in this situation, you're confronted by a person with cancer. You have to do something, so you get the cancer under control. Then you have to do something else, like a bone marrow transplant. But if you don't have a donor, you have to figure out a way to work around that. And then you just run into this cascade of problems that you have to address. And each problem, in the worst case scenario, which unfortunately is what this person experienced, each time you try to address one problem, it creates another problem. But I think that realistically, medicine is a process of doing things until there is simply nothing left to be done. In sort of the grand scheme of things, I think that our model of medicine that exists today is an active transition between illness and health. And in its idealized form, you are sick, you receive medical care, and you become healthy. But really, the purpose of medicine and the social function of medicine is to offer some kind of activity that will potentially restore health, or that will potentially mitigate illness. And the larger social implications of it are often lost in this kind of push to get from the sickness to the state of health. There's a much more idealized form of this process. This is an illustration from a book that was published at the University of California in San Francisco on clinical research. And this is one of my favorite illustrations, because down here on the bottom, it's explaining how to design a clinical research study. And then the clinical research at some point transcends this dotted line. And the ultimate goal of research, according to this textbook, is truth in the universe. That is within one school of thought, that is the ultimate goal of medicine, is to construct and utilize universal truth. And it's kind of funny, as someone who's interested in medical history, because that tends to never be the way that medicine unfolds. The person who actually developed the technology for bone marrow transplantation was named Edenal Thomas. He won a Nobel Prize for his work in transplantation in 1990. And at the international meeting of bone marrow transplant specialists every year, the keynote address is called the Edenal Thomas Lecture. So he casts a long shadow. But in his Nobel lecture, he gave an overview of his interest and where it came from. And in the 1950s, he said that the Atomic Energy Commission was essentially looking for medical techniques that would allow people to reconstitute their marrow in the event of a nuclear attack or a reactor meltdown. And so his initial experiments were in irradiated yeast, and then he expanded from there into dogs and eventually into humans. But there was no, there's obviously reality and science and truth in all of this, but this was not work that descended from a an abstract need for universal truth. This was science that developed within a very specific cultural milieu in the mid-20th century United States, out of a specific cultural anxiety regarding nuclear fallout that then developed into a separate, or coincided, I should say, with a separate cultural anxiety regarding the treatment of cancer. And here we are all these years later, still trying to grapple with the full ramifications of this technology that we have access to. The other thing is that even if medicine is at its most rational, that does not transcend social meaning. This was just a month ago in the New York Times. There was an article on stem cell therapy for sickle cell disease. And that's sort of where I think the field is heading increasingly, is that to navigate around the obstacles to transplantation, sickle cell disease is a life-limiting illness often that is vastly overrepresented in Black people in the United States. And again, transplantation or stem cell transplantation for sickle cell disease can be curative, but it's extremely risky, or can be extremely risky, I should say, because of the limited availability of donors. So the use of gene therapy or genetic modification of a person's own bone marrow is the current sort of technological frontier to work around this problem and provide care. At a very practical level, this New York Times article specifies that the gene therapy is extremely expensive and who's going to pay for it. But there's an even bigger, social implication than that of, what it means when you're doing genetic modification and when you're attempting to introduce a completely new technology to address a socially constrained area of inequality within medicine. You're ultimately shifting the risk of that new technology onto the very people that you're trying to help. And so in 10 years, what I think we're all optimistic about, what I'm optimistic about, is that gene therapy is going to be one of these great discoveries that, or great developments of medicine that made people's lives better. But the corollary fear to that is that if it turns out that this is not a long-term safe solution to the problems that we want to solve, we will have lumped all of that risk of a new technology onto people who were vulnerable to begin with. And the origins of that vulnerability are the things that I've been talking about. So in larger terms, I think the question that this brings up for me, and it's a much bigger question than just stem cell transplantation, but we don't have a good way of talking about congruence between social structures and genetics. I think that when those things diverge from each other, it's relatively straightforward to say that social structures are purely constructed and that genetics is a separate or indifferent or objective science. But I don't think we have a good way of talking about how these social structures and biological information intersect with each other and how we navigate that intersection. How we define the social meaning of genetics and what we do when our social structures appear to be reinforced by biological data. I think that's a very socially fraught area, and as genetics begin to influence more and more of our lives, this is just going to be a recurring problem. In medicine, we tend to, again, use technology to jump past these things, but the larger implications of it are going to still be there. Within that, there's this concept of genetic vulnerability, and it's a very concrete concept when you talk about an illness like sickle cell disease, where we can point to the gene that's mutated and the result of that mutation in the red blood cells and then the disease that results from it. But again, genetics is such an expansive concept, and we have such an easy way of slipping from conversations of genetics to other conversations that are socially constrained. We're just going to be seeing more and more applications of genetics in society. And especially as we enter potentially a technological future where we can change people's genetics, there are significant implications to all of this in terms of which traits we consider, or which genes, I should say, we consider to be beneficial and which ones we consider to be subject to change. And so as we're moving forward, again, I think the technology will eventually evolve in such a way that HLA matching is going to diminish in its importance over the generations. But the larger question of how we have structured our world around traits and how those traits interface with genetics and biology is going to be an ongoing one. So on that note, I will conclude, and I am happy to take questions.
Mike, thank you so much for this. I am just already struck by the really complex story that you're telling, and the range of evidence and sources that you're combining here, right? Individual case studies, history, obviously medicine and science, but also thinking in terms of rhetoric and narrative. So incredibly generative. Thank you.
Thank you.
So yeah, folks, what questions do you have? You're welcome to type them into the chat. You can use the raise hand feature and we can call on you. We'd love to get this conversation started.
I see that somebody read the same article I did recently about the land bridge migration story being questioned. And that is, you know, when I alluded to those arrows being produced by sort of a common narrative that's emerged from anthropology and archaeology rather than this sort of objective or indifferent concept of genetics, I think that illustrates that point, that our concept of our origins, our understanding of genetic data is mapped onto our understanding of our social origins and not necessarily the other way around.
Other questions or comments, or perhaps answers to some of these incredibly generative questions about next steps in this field? Well, as folks are thinking and typing, Mike, I'd like to hear a little bit more about how you communicate these kinds of, or how you even frame these kinds of questions for an audience that maybe is more motivated by that idealized version of medical research, right? To go from research question to universal truth, especially if as you say, the goal is to, come up with a technological fix without actually asking or grappling with the nature of the problem itself. I'm just wondering, what strategies have you been using to do that?
That's an excellent question, and one I do not have a great answer to. My kind of plan is to putting this talk together was a great opportunity to get my thoughts in order and make something cohesive. So what I'll probably end up doing is submitting a version of this talk to a medical journal and just kind of see what they make of it, if they drop this, if they refuse it outright, or if they have some questions. This thing that I call medical pragmatism, it's I would say the core of what we do in medicine in the United States in 2021, which is that you use technology to solve problems. And there's a lot of power in doing that. I mean, the fact that most of us expect that if we get sick, we'll get better is informed by relatively new technological technologies that have been available for maybe a few generations. So the power of that is so self-evident that I think it becomes very difficult to talk about the larger implications of it, because those don't have any practical applicability in terms of making a sick person healthy, but they have tremendous implications in terms of our overall role in society and the place that medicine fits. I think that this particular topic is also especially challenging because anytime that you talk about the intersection of race and especially about systemic inequality in medical care, people can get very nervous that they are being accused of personally being racist or of intentionally participating in a racist system. And the journal articles, for example, that I was citing here, I didn't pick them because they're bad articles or because they don't convey good information. I picked them because they're very good articles. They were very influential, and they present a clear piece of information in an interesting way. And the unquestioned assumptions that inform those articles are very interesting once you take a step back and actually think about them. So I think the implications of this for medical practice are potentially to clarify some of our understanding of clarify some of our assumptions that we're making that aren't generally questioned, and also to diversify the kinds of knowledge that we would consider to be valid within medicine. Then somebody is asking in the chat, I mentioned the influence of nuclear families on the initial transplant studies. The earliest transplants were done in the late 1960s, and they were exclusively done between siblings who were matched to the greatest extent possible with the technology at the time. It was a slightly different set of tools that they would use. But the very first transplant ever was at the University of Minnesota for a baby who had SCID and was born without an immune system, using, I believe, the baby's brother or sister. And the first transplant in an adult was by Edenal Thomas in Seattle, using a person with leukemia and his brother as the donor. And until this is an area where I wish I could be a little bit more firm, but the introduction of newer immunosuppressive medications and also the increasing recognition of HLA genes and their function, which happened in the 1980s and 1990s, sort of broadened out the availability of donors. But really, outside of a sibling context, it was very unusual to do bone marrow transplants, half a century ago.
Other questions or comments for Dr. Winstead? Oh, it looks like we've got a question in the chat for you from Jesse, who writes, I loved the way you expressed the ineffectiveness of our language to address points of congruence between social and genetic categories. Not to put you on the spot, but here we are putting you on the spot. But especially as a practicing clinician, do you have any suggestions on how we approach those convergences when in dialogue, especially with patients?
Yeah, this is something that I grapple with, honestly. And part of the reason is that the process for identifying a donor is the same. Your likelihood of finding a donor changes depending on the demographic group that you belong to and whatever interaction with genetics that has. But we go through the same process, and then we either find a donor or we don't. The challenge is that I don't, and I am open to other opinions on this. I personally don't like to tell people that the odds are bad going in. I prefer to go through the process, see if there's a donor available, and then have a conversation about what we do when there isn't a donor available. That's the practice that I've developed. I've worked with people over the years who would sometimes caution families, especially if the family was Black or if the family had parents of two different demographic groups, that their likelihood of finding a donor was going to be lower. But I'm not sure what the productivity is. And I think the anxiety that gives me is that you're implying something about the structure of this family or about this person's identity, that it's somehow their fault that if the search doesn't turn out well.
Excellent. And we have another question in the chat about how race is addressed in the medical literature itself. So Kantia writes, can you expand on how race is treated as a scientific construct and or a social construct within the literature? Is race still treated as a biological reality, even though race has been accepted as a social construct only? How does the literature navigate that?
Yeah, race is it's a very fraught notion in the medical literature. And I apologize if I go on with this. But one of the reasons that the medical literature in the 21st century tends to be so concerned with race is the long-term fallout of racist practices in medicine from the 19th and 20th centuries, whether that was the neglect of African-American people in clinical studies or the abuse of African-American people by physicians in the context of clinical studies and clinical practice. And so the trouble is that an awareness of race is generally considered mandatory in the conduct of a clinical trial and in the conduct of medical science in order to address those systemic inequities and the abuse that was experienced by Black people. the problem is that when again, your model of the data that you're generating is founded in the concept of universal truth, it's very easy for this kind of public health orientation and this interest in equity to take on the character of biological essentialism, or to suggest that you're demonstrating something essential about a person when you show differences in a study based on race. And then the nature of those differences also plays into it. So kind of the classic example from when I was a medical student was that they found that certain blood pressure medications worked better in people who identified as Black, and other blood pressure medications worked better in people who identified as white. And there was a cultural commentator at the time who said, I have a white mom and a Black dad. Like, does the drug work 50% better for me or 50% worse? Which is, kind of gets at the problem that you run into when you start saying that there are disparate treatments that are going to be targeted toward demographic group. Part of that is that you identify demographic group through this combination of social cues and self-identification, and those things are not necessarily biological. And then as soon as you start tying those to things like you have to take a few kind of fuzzy logical leaps in order to tie those to biological reality, like the efficacy of medications or other things that we observe. And so I think the kind of short answer to your question is that depending on the context, the medical literature will either discuss race as a biological determinant or as a social construct. And it really depends on the question that is being asked, and the implications of that are kind of obvious when we talk about it, but they tend to be overlooked.
Well, we are right around 4:30, or actually a couple of minutes over 4:30. So I want to pause again and thank everyone for being here, and especially for Dr. Winstead for joining us this afternoon and sharing these really important questions with us. So more to consider, and we can continue to think about these things. If you will bear with me for just a moment as I plug the rest of this series, we have another speaker joining us later this month on the 27th. Christina Gupta from Wake Forest University will be talking about part of her book, Medical Entanglements, especially related to transition-related care in a sort of pre and current COVID context. So I hope you'll join us for that. You can join our mailing list and hear all about these talks to come. So one final thank you to everyone for joining us, and especially thank you to Dr. Winstead for this generative talk.
Not to do if you are approaching the question of school performance and with the belief that it's genetically determined. It's just she keeps coming up in different publications.
Oh no, that's so funny to have that person in mind. That's like, okay, it's not that. This is not how we do.
Yeah, exactly.
Whatever I'm doing, it's not that.
Well, in this perception that genetics are objective and that the science is not socially embedded is a very appealing one, I think. And
totally, yeah, because I don't want to say absolves you of that history, because that's not quite what I mean, but you don't have to think about it or grapple with it or ask these more foundational questions about the problem in the first place. If, like you said, you can come in with the technological fix that's like, okay, genetic test or some kind of like genetically based treatment, let's move on and not consider it. So, and gosh, your justice questions or points about extending the risk to the folks who are already the most vulnerable, I just think is a really important question and call toward health justice. So
yeah, it's challenging. Sorry, I won't keep you all.
Yeah, I'm just, all of this is to say it was so generative, and I have so many notes and so many ideas that it was great.
Well, next time you have me back, I'll try to have a closed loop kind of type of thing, but we're not there yet. All right. Well, again,
how do you address all of this in one talk, let alone one career? So it's a really great start. I'm excited.
I thank you so much. This was a huge pleasure, and honestly, this is the most nervous I've been giving a lecture in a long time.
A totally different kind of audience. Yeah.
Okay. Well,
thanks again. I really appreciate it.
Yeah. Have a good day. Thank you very much, Irma.
Yeah, great to see you. Bye-bye.